Thank you for your interest in my poster entitled Development of a Hybridization LCMS Methodology for Quantitation of siRNA in Cynomolgus Monkey, presented at the OTS 2024 in Montreal, Canada. My name is Jean-François Dupuis, Senior Research Scientist, Method Development at Altasciences, and I will give you a brief overview of the work we have conducted. siRNA are short, double-stranded oligonucleotides that can regulate gene expression by inducing mRNA degradation. Hybridization LCMS-MS has been successfully used in the past years for the quantitation of single-stranded oligonucleotides in matrix. This approach relies on capturing the oligonucleotide of interest with a full-length complementary DNA probe, followed by analysis by LCMS-MS. Applying the same workflow to a double-stranded species presents new challenges as the capture probe competes with the sense strand of the siRNA. Here, we present the first adaptation of the existing hybridization workflow to quantitate the double-stranded siRNA1. In the hybridization workflow, a full-length complementary DNA-based probe is used to capture single-stranded oligonucleotides because of its high specificity and affinity. Using a specific DNA probe, very good recovery of the antisense strand of siRNA was achieved as demonstrated in the left figure. However, the figure on the right shows that, under the same conditions, recovery of the double-stranded siRNA was poor at lower temperatures and high salt concentrations due to the competition from the siRNA1 sense strand. To overcome this competition, a peptide nucleic acid, or PNA, probe was tested. The peptide nucleic acid probe offers a significant advantage due to the absence of the negatively charged phosphate backbone, which eliminates electrostatic repulsion with the siRNA antisense strand during the annealing process. Moreover, the higher melting temperature of the PNA-DNA complex allows for efficient annealing at higher temperatures. Under conditions of 65 degrees and 10 millimolar sodium chloride, a recovery of 90% was achieved using the PNA probe, compared to 55% with the standard DNA probe. Using the PNA probe in the hybridization workflow, the assay showed very good precision and accuracy over the range of 2 to 1,000 nanogram per ml. In conclusion, a bioanalytical assay was successfully developed for the quantitation of a double-stranded siRNA using a PNA probe. The method was also transferred to monkey CSF and tissues and was used for non-GLP sample analysis.